Hello, my name is Muhammad Nafis bin Afzal Nizam with matrix number AW220202. Want to present about a poster that based on the thesis that titled Biosurfactant Production by Lactic Acid Bacteria that isolated from homemade vegetables pickle. Firstly, I would like to introduce a bit introduction about the topic of research from the thesis. Biosurfactants or bioemulsifiers are surface-active compounds produced by diverse microorganisms at the microbial cell surface or excreted extracellularly. They can be divided into two basic groups, which are synthetic surfactants and biosurfactants. Synthetic surfactants are chemically synthesized through organic chemical reactions, while biosurfactants are excreted by microorganisms such as bacteria, yeast, and fungi. Biosurfactants have special advantages over synthetic surfactants such as biodegradability, pH, and salinity. Bacteria are the main group of organisms generating biosurfactants to microorganisms such as Pseudomonas species, Acinetobacter species, Bacillus species, and other various types of bacteria. In scientific research, they are among the most commonly studied bacteria. The aim of the study is to characterize and evaluate the biosurfactant production of the isolated lactic bacteria from the homemade vegetables pickle. Based on the introduction earlier, we can identify a few objectives from the topics from the thesis. Firstly is to identify efficient biosurfactant producing lactic acid bacteria strains from homemade pickled vegetables. Next, to assess the antimicrobial activity of biosurfactant producing probiotic lab. Lastly is to investigate the probiotic and biosurfactant properties of lab isolated from homemade pickled vegetables. In the thesis, there are few methodologies that be used. So there are 4 of them that have been chosen from the thesis. Firstly is pH tolerance. Next is sodium chloride tolerance, all spreading technique, and lastly is antimicrobial activity. Based on the result from the thesis, it shows that the isolated lactic bacteria can grow in a different concentration pH. From the result from the thesis, it shows all the samples of the lactic acid bacteria show a positive result. This shows that all isolated bacteria can resist the acid condition under the pH 1, 3, and 5. Next is sodium chloride tolerance. Based on the result from the thesis, it also shows all the samples of lactic acid bacteria shows positive result from the 3% and 5% concentration of sodium chloride. From the result, isolated acid bacteria, it shows that have the tolerance to the high concentration of sodium chloride, which is 3% and 5%. It also indicates that it managed to survive under harsh conditions. From the result from the thesis is the determination of biosurfactant production by using oil spreading technique. This technique is used to determine the presence of biosurfactant production. Based on the result above from the thesis, it concludes that all isolated bacteria show the positive result, which is all positive measurement 3, 4, 4, 5, 4.5, 6.5, 6.7, and 5.5 cm, which indicate result which all have biosurfactant production. Lastly is the antimicrobial activity. It shows the inhibition zone against the Escherichia coli and Staphylococcus. From the result, it shows all isolated bacteria shows positive result, which inhibition zone are above 12 mm. But the best result get from the experiment is C3 and G6 from the thesis result. Based on the result obtained, the discussion from the thesis. The diversity of biosurfactant producing microorganisms and of the biosurfactant produced is well known. So in this experiment, we screen the probiotical potential by letting the isolated bacteria grow in low pH levels and high concentration of sodium chloride. The result we got is the isolated bacteria show positive result, which has tolerance to low pH values and sodium chloride. Next, to test whether the isolated bacteria contain biosurfactant production, we use oil spreading technique. If they contain biosurfactant production, the oil will form a clearing zone. So from the result, all the isolated bacteria contain biosurfactant production. Lastly, isolated bacteria have the potential to inhibit the growth of bacteria. In conclusion, 8 lactic acid bacteria were successfully isolated according to the data obtained. It consisted of all isolated bacteria as gram-positive bacteria. The isolated bacteria had capacity for probiotics such as low pH and salinity. Antimicroactivity. Use 3% molasses as a renewable base. This isolated can generate biosurfactant. In a study, the most potential producer of biosurfactant was found to be all bacterial isolates identified as lactic acid bacteria. That's all from me. Thank you.